Hello everyone, welcome to our own YouTube channel Shantanu Kolhe Lifescience. So today we are going to discuss glycolysis process in detail. In the last video we have discussed all the concepts and basics of metabolic pathways. In order to understand metabolic pathways, you need to understand the concepts and basics first, which we have discussed in the previous video lecture. If you haven't watched that video, make sure you watch it so that you can understand the further things in detail very easily. Okay, let's start with the glycolysis pathway in detail with the help of structures. Starting with the salient features of glycolysis or characteristic features of glycolysis. The first point is that glycolysis is also known as EMP or Emden-Meyerhoff-Parnas pathway. Because of these three scientists, the name is also the name of glycolysis is also EMP pathway. Okay. Glycolysis is derived from Greek word glucose means sugar and lysis means dissolution or breakdown. Location of glycolysis is that it occurs in cytosol in both prokaryotes as well as in eukaryotes. It can occur in aerobic as well as in under anaerobic condition. If it occurs in aerobic condition, the end product is pyruvate. In glycolysis, the glucose will be converted into glucose will be broken down into pyruvate, and this pyruvate will be further broken down or oxidized into carbon dioxide and H2. Oxidation of pyruvate into carbon dioxide and H2O, we will cover it in upcoming videos. If the glycolysis occurs under anaerobic condition, the end product which will be formed is lactate. It can occur in tissues lacking mitochondria. This point is very much important. Example is RBCs, that is red blood cells, cornea, lens, etc. These points you have to remember because there is no trick for it. Okay. The intermediates of glycolysis are used in the synthesis of amino acids and fat. Reversal of glycolysis at irreversible steps is known as gluconeogenesis. Irreversible steps means the change in free energy is large negative, and this is the reason why the reaction will favor in forward direction and not in backward direction, which we have discussed in the previous lecture. And irreversible steps are regulatory in nature. So if we bypass those irreversible steps, we will get again glucose. That is, there will be formation of glucose. In glycolysis, there is breakdown of glucose. But in gluconeogenesis, when we bypass those irreversible steps, we will move towards glucose. And thus there will be formation of glucose. We will understand it when we will study gluconeogenesis. Okay. So there are three phases of glycolysis: energy investment, priming stage, and preparatory stage. As the name suggests, energy investment, which means there will be utilization of energy in the form of ATP. Splitting phase, during which one molecule of 6 carbon compound will be broken down or will split into one molecule of 3 carbon compound and second molecule of 3 carbon compound. The name of this 6 carbon compound is fructose 1,6-bis phosphate. This fructose 1,6-bis phosphate is a 6 carbon compound which will break down into one molecule of dihydroxyacetone phosphate of 3 carbon and one molecule of glyceraldehyde 3 phosphate of 3 carbon. Okay, we will study that when we will actually look that slide. Okay, the next phase will be energy generation or payoff phase. As the name suggests, energy generation, which means there will be formation of ATP or production of ATP. So now we will start glycolysis steps one by one. Okay, glucose will be converted into glucose 6-phosphate, which will be catalyzed by an enzyme hexokinase, which require magnesium and manganese ions to function efficiently. During this step, there is consumption or utilization of one ATP. One ATP is used. This step is known as phosphorylation. Most of the phosphorylation steps are irreversible steps, and irreversible steps are regulatory steps. There are certain or rare exceptions which we will study in upcoming slides. Which is that exception, we will study that. Now, one more important thing is that if you only remember the structure of glucose and fructose, you can draw any intermediate and metabolites of the glycolysis pathway. You can draw their structures very easily. Only you have to remember glucose and fructose structures. Okay. Even if you don't remember ring structure, it is okay, but you have to remember linear structure of glucose and fructose. Only two structures you have to remember. Okay. I will show you the trick by which you can easily deduce all the structures of the metabolites or intermediates of the glycolysis pathway. This is a glucose molecule. Glucose is converted into glucose 6-phosphate. 6-phosphate, which means, according to the name, if you logically think, 6-phosphate means there should be an addition of phosphate group at 6 position or 6 carbon position. As you can observe here, at 6 carbon position, there is addition of phosphate group. Addition of phosphate group, and thus the step name is phosphorylation. You also have to remember that kinase, the role of kinase is the addition of phosphate. Okay. This is linear structure of glucose. You can observe that at 6 carbon position, there is addition of phosphate group. That's why the name has been changed to from glucose to glucose 6-phosphate. Easy. Now, this is a structure of fructose, and this is a linear structure of fructose. You just have to remember glucose and fructose structures. Okay. There is also one trick by which you can remember half of the structure of glucose and fructose. In fructose, carbon number 3, 4, 5, 6 is same as that in glucose. Okay. In glucose, at carbon number 1 there is aldehyde group. Carbon number 2 you have to remember. Here, first and sixth position has CH2OH. Second position has ketone group. So by this trick you can remember glucose and fructose also. Only these two structures you have to remember. Rest all of the structures you can draw. The second step is glucose 6-phosphate will be converted into fructose 6-phosphate, which will be catalyzed by an enzyme phosphohexose isomerase or phosphoglucoisomerase. It requires magnesium ions to function efficiently. This step is known as isomerization, and it is reversible step. So why this step is known as isomerization? Because glucose 6-phosphate and fructose 6-phosphate have same chemical formula. Only there is difference in the arrangement in chemical structure. Okay. So this is glucose 6-phosphate. At 6th position there is addition of phosphate group. This is fructose 6-phosphate. At 6th position there is addition of phosphate group. This is linear structure of glucose 6-phosphate. At 6th position of glucose there is addition of phosphate, converting glucose to glucose 6-phosphate. At 6th position of fructose there is addition of phosphate group, converting fructose to fructose 6-phosphate. If you actually count the number of carbons, hydrogen, oxygen, you will find that it is 6, 11, 6 respectively, and phosphate group also. Okay. In the same in fructose 6-phosphate, if you count, you will find these values. Okay. So both of these numbers, they will match. Okay. So they are isomers because they have same chemical formula but difference in the arrangement in chemical structure. So they are isomers of each other, and hence the step name is isomerization. Okay. And the name of enzyme is also isomerase. So you can get idea from that also. Okay. Third step is fructose 6-phosphate will be converted into fructose 1,6-bisphosphate. Here also 1 ATP will be used. It is catalyzed by an enzyme phosphofructokinase 1. It requires magnesium ions to function efficiently. This step is phosphorylation. Okay, it is, as I said earlier, most of the phosphorylation steps are irreversible in nature, and irreversible steps are regulatory steps. There is one example or rare exception which we will study in upcoming slides. Okay. Now, fructose 6-phosphate, there was an addition of phosphate group at 6th position. Fructose 1,6-bisphosphate. 1,6. 1 is also added now. So there will be addition of phosphate group at first carbon position also, and 6th carbon position it was already there. So the name has been changed from fructose 6-phosphate to fructose 1,6-bisphosphate. You can observe it from here also. Fructose 6-phosphate. Now there will be addition of phosphate group at first position also, at first carbon position also. So the name has been changed from fructose 6-phosphate to fructose 1,6-bisphosphate. Similar. It is very easy to remember if you only remember glucose and fructose chemical structures. Easy. Fructose 1,6-bisphosphate will split or it will undergo cleavage to form two molecules of 3 carbon compound each. So 6 carbon compound will be broken down into one molecule of 3 carbon compound and second molecule of 3 carbon compound. 3 plus 3 equal to 6. So that is splitting. So this step comes under splitting phase of glycolysis. It is catalyzed by an aldolase enzyme which requires zinc to function efficiently. Now, even if you don't need to remember DHAP and glyceraldehyde 3-phosphate structure separately, you can observe that from fructose 1,6-bisphosphate, if we split it from the middle, that is after third carbon, if you write down the same as it is, you will get the structure of dihydroxyacetone phosphate, that is DHAP. And the lower half, if you write down the same, it will be glyceraldehyde 3-phosphate. Glyceraldehyde means there will be aldehyde group for sure, which is present in first position or first carbon. And 3-phosphate means phosphate group will be present at third carbon position. So here you can observe phosphate group at third carbon position. So you can make these structures very easily. Okay. In this step, there will be interconversion of DHAP into glyceraldehyde 3-phosphate, which is catalyzed by an enzyme phosphotriose isomerase. As the name of the enzyme is isomerase, the step is isomerization, which is a reversible step. There are two isomers which have been formed, DHAP and glyceraldehyde 3-phosphate, as evident from their chemical formula. If you actually count the number of carbon, hydrogen, oxygen, you will find that in DHAP and glyceraldehyde 3-phosphate, they will be same. Now, there will be two molecules of glyceraldehyde 3-phosphate. How? In this, originally one glyceraldehyde 3-phosphate molecule will be formed, but this DHAP will also interconvert into glyceraldehyde 3-phosphate. Means this molecule was already there, and if this DHAP convert into glyceraldehyde 3-phosphate, there will be two molecules of glyceraldehyde 3-phosphate. Okay, that's why we write two molecules of glyceraldehyde 3-phosphate. Now this step, most of the students get scared of this step, that is 6th step. It is very easy. Okay, two molecules of glyceraldehyde 3-phosphate will be converted into two molecules of 1,3-bisphosphoglycerate. It is catalyzed by an enzyme glyceraldehyde 3-phosphate dehydrogenase. This step is oxidation and reversible. Why is it oxidation? In this enzyme glyceraldehyde 3-phosphate dehydrogenase will remove hydrogen ions and electron from glyceraldehyde 3-phosphate, and it will pass it to its coenzyme, that is NAD+. NAD+, it is in oxidized form. You need to understand two terms. Oxidation. Oxidation means removal of hydrogen ions and electrons is known as oxidation. Acceptance of hydrogen ions and electron is known as reduction. So if this is positive, you can logically answer that if it is positive, there will be loss of electron in it, which means it is oxidized. And when this oxidized form, that is NAD+, will accept hydrogen ions and electron, that is reduction is taking place, NAD+ form is oxidized, NADH form is reduced form. Okay. Oxidation of aldehyde and to carboxylic acid, this we will understand while studying its structure. Okay. Now you need to understand one thing, this NAD+ or NAD positive is in oxidized form. It will accept hydrogen but not directly hydrogen. It accepts hydrogen in the form of hydride ion. Okay, this hydride ion is H+ plus 2 electron. So if it accept, it will form as NADH, and this free hydrogen ion will come from inorganic phosphate. We will study it when we will understand structure in the next slide. So if there are two molecules of glycerolated 3-phosphate, it will accept two hydride ions. One hydride ion is H plus 2 electron. Two hydride ion will be 2H plus 4 electron. It will get converted into 2 NADH, and 2H plus will come from inorganic phosphate. Okay, that's why this is the way of writing it, NADH plus H plus. Okay, glyceraldehyde 3-phosphate. There is removal of hydrogen atom, which means oxidation is taking place, and addition of phosphate group at first position. Name will change from 3-phosphoglycerate, glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate, because now there is addition of phosphate group at first position also, and addition of phosphate group at third position also. So the name has been changed from glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate, and there is removal of hydrogen atom also. Okay, so you can easily deduce this structure. Okay, next, two molecules of 1,3-bis phosphoglycerate will be converted into two molecules of 3-phosphoglycerate. Okay, two ATPs will be formed in this process, which we will understand this, we will study when we will study the structure in the next slide. It is catalyzed by an enzyme phosphoglycerate kinase, which require magnesium ions to function efficiently. This step is phosphorylation. Now, as I was saying, this is a rare exception, because mostly phosphorylation steps are irreversible in nature and irreversible steps are regulatory in nature. But in spite of this being a phosphorylation step, it is reversible step and it is not regulatory step. So this is an rare exception. That's why I have marked 4 stars over here, because it is important. Okay, now we will understand why is it known as phosphorylation. Because there is, you will ask one question, that if it is 1,3-bis phosphoglycerate and we are getting 3-phosphoglycerate, there is no addition of phosphate group, then why we are naming it as phosphorylation step? So we are calling it as phosphorylation step because this phosphate group will combine with ADP. We know that ADP plus inorganic phosphate, we will get ATP, and that is what happening over here. This phosphate group will combine with ADP to form ATP. From one molecule, one ATP will be formed, but since there are two molecules, two ATPs will be formed. That's why two ATPs are mentioned here. Okay, then after removal of phosphate group, as it is the structure which is left behind, you have to write as it is. You will get 3-phosphoglycerate. You don't need to remember every structure. Okay, I think now you are getting how to draw the structures. Only you have to remember glucose and fructose structures. Two molecules of 3-phosphoglycerate will be converted into two molecules of 2-phosphoglycerate. It is catalyzed by an enzyme phosphoglycerate mutase. This step is known as isomerization, because isomers are formed. 3-phosphoglycerate and 2-phosphoglycerate have same chemical formula. Only there is difference in the arrangement in chemical structure. Here is the phosphate group was present at third carbon position, but in 2-phosphoglycerate, as the name suggests, 2-phosphoglycerate, which means from third carbon position, the phosphate group will be transferred to second carbon position. That's why the name is 2-phosphoglycerate. So you can also make structure from here. Next step. Two molecules of 2-phosphoglycerate will be converted into two molecules of phosphoenol pyruvate, which will be catalyzed by an enzyme enolase, which require magnesium ions or manganese ions to function efficiently. This is a dehydration step, means there is a removal of water molecule. Peptide bond formation is also a dehydration step, because there is a loss of water molecule present. Okay, 2-phosphoglycerate. From 2-phosphoglycerate, there will be removal of H2O. So OH will get removed from third carbon and H will be removed from second carbon. The structure which is left behind is this. As it is, you have to write. You will get the structure of phosphoenol pyruvate. Again, you don't have to remember phosphoenol pyruvate structure separately. Okay. And this is the last step of glycolysis. Two molecules of phosphoenol pyruvate will be converted into pyruvate. Now, very less books have this information that phosphoenol pyruvate is not directly converted into stable pyruvate form. First, it will be converted into enol form, which is unstable. Okay, it is catalyzed by an enzyme pyruvate kinase. It requires magnesium and potassium ions to function efficiently. This step is phosphorylation. We will understand it in the next slide why. It is irreversible and regulatory step. Only the exception was 7th step. In spite of being a phosphorylation step, it was reversible and not regulatory step. Now, how two ATPs are getting formed, we will understand it. Okay. You can observe this phosphate group will combine with ADP to form ATP. But since there are two molecules, we will get two ATP. As simple as it. First, we will get unstable enol form. If the molecule is unstable, it will try to get stable as early as possible or as fast as possible. Thus, the process is spontaneous. And the conversion of enol form to keto form of pyruvate is known as tautomerization. Now, you will have one doubt. Here CH2, above CH2 there was one single bond. Here there are two double bonds. Why? There is change in the number of bonds. You need to understand one thing. Carbon can form maximum 4 bonds. So, if we see over here, CH2 means I can write it like this. And if we count the number of bonds, it will be for this carbon, these 2 bonds, this is 3rd bond and this is 4th bond. 4 bonds are there. For this carbon, this is 1, 2 and these 2 bonds will be 4. Okay. Now if it would have been a single bond, here it is single bond, then this carbon would have formed only 3 bonds, 1st, 2nd and 3rd. But it is incorrect because carbon have to form 4 bonds. Even if you count number of bonds for above carbon, for this carbon are bond number 1, 2nd and 3rd. So this is wrong way. That's why number of bonds here are 2. Okay. Okay. After tautomerization, you will get this keto form. Okay. In this, now you will say that there is only single bond over here. So if I draw a structure, it will be, if there were double bond like this, then it was not correct because these 2 bonds, 3, 4, 2nd, 3rd and this is 5th. But carbon cannot form 5 bonds, it can form 4 bonds. That's why there is a single bond over here. So if you get a question that pyruvate structure is in option, if you get pyruvate structure like this with double bond, this will be incorrect option. Okay, you have to count the number of carbon, you have to count the number of bonds which the carbon is forming. Okay. So we are done with the glycolysis steps. Just with the help of, or just after memorizing glucose and fructose structure, you can deduce or you can make all of the chemical structures by yourself with just remembering glucose and fructose structures. Okay. Now we will see inhibitors in glycolysis. This is also very important slide because questions are asked from inhibitors. Iodoacetate is an inhibitor of glycerolide 3-phosphate dehydrogenase. It forms covalent derivative of SH group of enzyme active site and render it inactive. You just need to understand this one statement. You don't need to study this in detail, the mechanism and catalysis mechanism of this enzyme in detail, because we are students of life science and not of chemical science, because the catalysis mechanism is very, very difficult. Okay, and the question will never be asked from there. You need to understand that iodoacetate will form covalent derivative of SH group present on enzyme active site and it will render the enzyme inactive. Next point is fluoride inhibit enolase enzyme. It is a competitive inhibitor. If this is an enzyme with its active site, this triangular shape is active site. Okay, and if this is normal substrate, in normal case it can easily bind with the active site. Okay, and when there is competitive inhibitor also present, so this is normal substrate, and if this is competitive inhibitor which look same as normal substrate, this is inhibitor. Since they look very much same or they resemble in their structure, they can easily bind with active site. Inhibitor can also bind, normal substrate can also bind. If inhibitor bind, it will inactivate the enzyme. If substrate bind, it will render the enzyme active. But as you can see that there is competition for the binding with active site. This can also bind and this can also bind. So there is a competition. Thus, such inhibitor which resemble the normal substrate will be known as competitive inhibitor. Okay. Significance is that fluoride is added during blood glucose estimation in labs. It inhibits glycolysis and thus glucose will not be used up in glycolysis, due to which accurate measurement of blood glucose level is possible. Now let us understand this with interesting example. If this is a test tube, a patient want to estimate the blood glucose estimation. A patient want to get the reports of his blood glucose levels. If he goes in lab 1 and lab 2, he will get different results. Why? Suppose here in test tube, here is blood and blood has glucose. Okay. Suppose lab 1, according to their lab protocol, they don't add fluoride in their blood glucose estimation SOP, and in lab 2 they add fluoride. If the patient initially if the blood glucose levels in the patient is 100 mg per deciliter, after some time it will reduce, suppose 80 mg per deciliter. Why? Because this glucose will enter glycolysis, and we know that in glycolysis there will be breakdown of glucose into pyruvate. So ultimately the levels of 100 mg per deciliter glucose will reduce to 80 mg per deciliter and it will give faulty result. Faulty result it will give. But in lab 2, if initially the blood glucose level is 100 mg per deciliter, after some time also the blood glucose level will be accurate. Why? Because fluoride will inhibit glycolysis. If glycolysis is inhibited, glucose will not get converted, glucose will not get used up in glycolysis pathway. So that's why there will be accurate measurement of blood glucose levels in lab 2. The report will be best or the results will be best. Now I think you have understood the significance of fluoride. Easy? Okay. This slide is also very much important because questions are asked from here. Hexokinase is present in all tissues. Glucokinase is present in liver. Hexokinase enzyme has low Km value of 0.1 millimolar and glucokinase has high Km value of 10 millimolar. Hexokinase catalyzes the phosphorylation of hexoses like fructose, mannose, etc. Glucokinase catalyzes the phosphorylation of only glucose. Km means Km is inversely proportional to the affinity of enzyme for substrate. If Km value is low, affinity of enzyme for substrate will be more. If Km value will be high, affinity of enzyme for substrate will be lower. Okay. Hexokinase has low Km value. So hexokinase is an enzyme and substrate will be glucose. Okay. So hexokinase has low Km value, which means this enzyme will have higher affinity, higher liking or more attraction for substrate. It has more attraction or more affinity for substrate. So if it has more affinity for substrate, even if substrate is present in lower concentration, it can bind with the substrate can bind with the enzyme very easily, even if it is present in low concentration, because the affinity or attraction or liking of enzyme for substrate is more. Okay. Now glucokinase has high Km value. If the Km value is higher, the affinity of enzyme for substrate will be less. Enzyme is glucokinase and substrate is glucose. If the affinity or liking or attraction of enzyme for glucose is less, the substrate would have to be present in higher concentration to actually bind with the enzyme. Why it need to be in higher concentration? Because the affinity or liking or attraction of enzyme towards substrate is less, because the Km value of enzyme is higher. So only when the substrate will be in higher concentration, it can bind with the enzyme. And this happens when after eating, the glucose levels will rise, that is, it can reach above 100 mg per deciliter. And this is the reason why after meal, glucokinase will work, and in rest of the cases, hexokinase will work. Okay. So I think you have understood this better. Okay. Moving on to the last slide of glycolysis. Energetics of glycolysis. In the first and third steps, one ATP was getting used. So 2 ATPs used. In the oxidation step, that is glyceraldehyde 3-phosphate dehydrogenase step, we were getting 2 NADH molecules were formed. When one NADH molecule passes through electron transport chain or oxidative phosphorylation, we get 3 ATP or 3 ATPs are formed. when 2 NADH molecule passes through electron transport chain, we will get 6 ATPs. 6 ATPs formed. 6 ATPs formed. In the 7th step, that is reversible step, in spite of being a phosphorylation step, it was reversible. In that step also 2 ATPs were getting formed. And in the last step, that is conversion of phosphoenol pyruvate to pyruvate, also 2 ATPs were getting formed. So total 10 ATPs formation occurred. 2 ATPs were used up in the energy investment stage, that is steps number 1 and 3. Resultant is 8 ATPs. So 8 ATPs are formed via glycolysis path. Very easy. Okay. I think this was the last slide and we are done with glycolysis. So we have observed that only from glucose and fructose you can draw all the structures. You don't need to remember or memorize all the structures of glycolysis intermediates or metabolites. Okay, we have also discussed inhibitors. Okay, and we have also discussed the significance of fluoride, hexokinase and glucokinase in detail. So there is nothing difficult in glycolysis. It depends on the quality of teaching, the quality of content preparation, because nowadays there are very less teachers who actually invest time to design the content, to study the content from different books, to modify the content in such a way that student can understand. They don't do it, they just copy and paste. And this is the reason why students are scared of such concepts, biochemistry and all. Nothing is difficult in this world. It depends on the teacher. Okay. In the reference book also, these structures were given in very different way, due to which students were actually scared of remembering these structures. But now you can remember each and every structure after watching this lecture. You just have to remember glucose and fructose structure. Okay. You have to practice previous year questions from glycolysis. After this, you will be able to solve any question from glycolysis. You can also solve tracing of radiolabel carbon question. For example, if you radiolabel, if the carbon number 1 of glucose is radiolabel, and if the question is asked that in the pyruvate structure, which carbon will be radiolabel, if carbon number 1 of glucose was radiolabel. So just by analyzing the structures or writing the structure on your own, just by remembering glucose and fructose structure, you can easily solve such kind of questions. And such kind of questions are asked in BARC exam, BARC OCES. I think in last year this question was asked. Okay, but there is nothing much difficult in such questions once you are familiar with the concept and once you strengthen your concepts. So the regulatory part, regulation of glycolysis part is left, which we will study it in next lecture, because I haven't added it in here because it will get difficult for you to understand everything. Okay, so in the next video we will understand the regulation of glycolysis. Okay, I hope you understood. See you in the next video. Thanks for watching.